In the world of precision oncology, we spend, a lot of time chasing predictive biomarkers. Like we sequence the tumor's DNA, we look for specific mutations, and we basically try to map out exactly how a patient will respond to a given therapy.
Right, yeah, that's the standard playbook.
Exactly. But when you step into the realm of immunotherapy, that predictive landscape gets incredibly murky. I mean, you can have two patients with virtually identical tumor genetics, give them the exact same immune checkpoint blockade, and then just watch one patient completely clear the cancer while the other doesn't respond at all.
Oh, it is. It's honestly one of the most frustrating and complex challenges in modern medicine. Because, the tumor's genetic profile simply doesn't tell the whole story. Immunotherapy isn't actually treating the tumor itself.
Where it's treating the immune system.
Exactly. You are relying on this highly dynamic, living network of cells to do the heavy lifting. And trying to predict the behavior of that network, it requires an entirely different level of biological surveillance.
And that is exactly what brings us to today's deep dive. So welcome, and we are so glad you are joining us for this. We are unpacking a massive, just groundbreaking study published in 2026 in the journal NPJ Precision Oncology. And the mission here is to decode a hidden predictor of immunotherapy response.
Yeah, and the scale of this research is just staggering. They didn't just look at, a handful of biopsies.
No, not at all. They utilized advanced single-cell sequencing to track the individual biological trajectories of nearly 80,000 T cells across 11 different patients with head and neck squamous cell carcinoma. Okay, let's unpack this.
So to really grasp this, we have to realize this deep dive isn't just about reading data spreadsheets. They essentially mapped the literal biography of an individual white blood cell, like tracking its journey from the blood across the vascular barrier and deep into the tumor architecture.
Yeah.
We get to see exactly how these cells alter their behavior before and after a patient receives treatment.
Right. So we know the play with neoadjuvant immunotherapy, or NIT. The clinical strategy there is to hit the patient with PD-L1 and TGF-beta inhibitors before the surgeon's scalpel even touches the tissue. The goal is to shrink the tumor early on and prevent micrometastases.
Yeah, neo meaning new and adjuvant meaning assisting, you do it first.
Exactly. But we also know the environment these immune cells are walking into, the tumor microenvironment, or TME, is notoriously hostile. Think of the TME as this toxic swamp, totally designed to confuse and tire out the immune system.
A toxic swamp is the perfect analogy. Tumors undergo aerobic glycolysis, the Warburg effect. So they consume massive amounts of glucose and aggressively churn out lactic acid. It creates an environment that is profoundly hypoxic, highly acidic, and structurally dense.
Wow, so they're literally starving the immune cells.
Yeah, exactly. When a T cell enters that space, it is physically and metabolically paralyzed. The tumor is intentionally hoarding the very resources the immune system needs to function.
Which actually brings me to a major conceptual hurdle in this paper that I need to ask you about. Because the authors focus heavily on driving the proliferation of exhausted T cells, or TECs. And wait, the paper talks about exhausted T cells as the goal. Why would we want exhausted immune cells? Like in any other organ, exhaustion is bad.
It does sound super counterintuitive, I know. But in the context of tumor immunology, exhaustion is actually a marker of extreme specificity.
Wait. Really? How so?
Well, think about a standard viral infection. Your T cells activate, clear the virus in a week, and then form a quiet memory pool. But a tumor doesn't just go away. The T cell enters that metabolic wasteland, encounters the tumor antigens, and experiences chronic, relentless stimulation of its T cell receptor, or TCR.
Ah, so it's fighting a literal war of attrition.
Precisely. And if a T cell stays maximally activated indefinitely, it would trigger a lethal autoimmune cascade. It would essentially melt the surrounding healthy tissue.
Oh, wow. So it has to calm itself down.
Yes. To prevent this, the cell adaptively upregulates inhibitory receptors on its surface. Proteins like PD-1, LG3, TM3, this is the exhausted phenotype. It's putting the brakes on itself. If we connect this to the bigger picture, when we see an exhausted T cell deep in the tumor bed, it's definitive proof that this specific cell has recognized the cancer and is actively fighting so hard it's burning out. They are the battle-hardened veterans.
That makes perfect sense. So neoadjuvant immunotherapy isn't magically waking up lazy immune cells. It's just chemically uncoupling those inhibitory brakes from the exhausted veterans so they can strike the final blow.
Exactly.
Okay, let's pause and map out where we are so far for you listening. Time for a quick recap. Bullet 1. Neoadjuvant immunotherapy primes the immune system before surgery by removing the brakes. Bullet 2. Exhausted T cells or TECs are tired because they are actively fighting tumor antigens in a toxic swamp. Bullet 3. Releasing those breaks lets the most specific veteran cells do their job. So keeping all that in mind, here's a question to you, the listener. If you were a general, would you rather have fresh troops who don't know the enemy or exhausted veterans who know exactly who to target?
That is the ultimate strategic dilemma, right? Because if you're an oncologist trying to predict if a patient will survive, who do you look at? The veterans already trapped in the swamp or the fresh recruits waiting in the wings?
Well, to answer that, we have to talk about how the researchers actually track these cells. Tracing an exhausted veteran versus a fresh recruit in a sea of 80,000 cells requires unbelievable precision. They used paired single-cell RNA and VDJ sequencing.
Yeah, the technology here it's just wild.
It really is. I like to think of it as a GPS and a diary. The VDJ sequencing acts like an immutable GPS tracker, a unique barcode for every single cell clone showing us where it goes.
Right, because when a T cell matures, it literally splices and shuffles its own DNA across the variable, diversity, and joining gene segments. That's the V, D, and J. The mathematical probability of two independent T cells generating the exact same receptor sequence is practically zero.
So you can look at a cell in the bloodstream on Tuesday, find a cell with that exact same barcode in the tumor on Friday, and know with absolute certainty that it is the exact same clone.
Exactly. And while the VDJ sequence is the GPS telling us where it is, the single-cell RNA sequencing the cRNA-seq acts as its transcriptomic diary.
Right, reading what the cell is feeling and doing right now.
Yes. By capturing the RNA inside the cell at the specific moment, we see exactly which genes are turned on. When the researchers read those cellular diaries, they found that before treatment, the T-cell clones were heavily compartmentalized. They were kind of stuck in rigid functional states.
But after the treatment, after the PD-L1 and TGF-beta inhibitors, that rigidity shattered, right?
Completely. The transcriptomic data showed massive phenotypic diversity emerging within individual clonal lineages post-treatment. The therapy gave the immune system back its ability to adapt and innovate on the fly.
Okay, time for another quick 5-minute recap of this segment. Bullet 1. Advanced single-cell sequencing uses VDJ as a permanent GPS barcode to track exact T-cell clones. Bullet 2. Single-cell RNA sequencing acts as a diary to read the cell's real-time behavior. Bullet 3. Treatment breaks T-cells out of their rigid, stuck states, allowing them to dynamically adapt to the tumor.
Beautifully summarized.
So, knowing we can read a cell's diary, here is my question to you listening. What specific trait do you think actually defines the cells that win the fight?
Well, and this brings us to the core mystery of the paper.
Yeah.
Because you'd think it's the veterans, right?
Right. But here's where it gets really interesting. The data revealed the true heroes of the story aren't just the exhausted veterans already inside the tumor. The critical predictors of survival are actually the fresh recruits coming from the blood. Enter a protein called granzyme K, or GZMK.
GZMK is such a fascinating biomarker. The researchers isolated this population of GZMK positive effector memory T cells, or TEM. These are cells that circulate in the peripheral blood. When we see high expression of GZMK, it marks that cell as a recent immigrant.
A fresh reinforcement that hasn't been burned out by the swamp yet.
Exactly. It's a highly capable progenitor cell stepping onto the battlefield.
But how do these specific cells know to leave the bloodstream and enter the tumor in the first place? I mean, they need an invite, right?
They do. The blood-based T cells heavily express a chemokine receptor called CXCR3.
Ah, so like a molecular VIP ticket to get into the club.
That's a great way to put it. When the tumor is inflamed, it secretes specific chemokines that create a chemical gradient. The GZMK positive cells in the blood use their CXCR3 ticket to detect this gradient, slip through the blood vessel walls, and traffic directly deep into the tumor bed.
But once they are inside, the diary changes, doesn't it? The paper details this massive biological shift.
It does. The in vitro studies in this paper perfectly captured it. When these fresh GZMK positive cells finally hit the cancer antigens and experience that intense TCR stimulation we talked about, they completely change.
They drop the name tag.
Yes. They immediately drop their GZMK expression, and simultaneously, they rapidly upregulate proliferation genes and those exhaustion markers. GZMK isn't the weapon used to kill the cancer, it's the signature of a progenitor standing on the edge of differentiation. The moment it engages the enemy, it transforms into an exhausted tumor killer.
Man, they are literal shapeshifters. All right, time to recap this commuter pathway. Bullet 1. Granzyme K, or GZMK, is a protein marking fresh, adaptable memory T cells coming from the blood. Bullet 2. They use the CXCR3 VIP ticket traffic into the tumor. Bullet 3, inside the tumor, they get stimulated, lose their GZMK name tag, and become the exhausted fighters that actually do the killing.
Perfect.
So question to you, if GZMK is a sign of a fresh recruit ready to transform, how could doctors use this information before ever giving a patient a drug?
That is the million-dollar clinical question. And to see how this plays out in a real human, we have to look at the actual spatial architecture of a tumor. Because tumors are not just homogeneous blobs of cancer cells.
Right. To visualize this data, the paper brings up patient 17. They use these incredible multispectral immunofluorescence micrographs.
Spatial context is everything. You have two main zones. First, the stroma, which is the connective tissue, the fibroblasts, the blood vessels.
So the stroma is basically the parking lot or the staging area outside the stadium.
Yes, exactly. And then you have the parenchyma. That is the dense nests of actual malignant cancer cells, the core battlefield.
Okay, so looking at patient 17, before treatment, they had a meager 10 proliferating GZMK positive cells per square micron in the parenchyma. The front line was empty.
But post-treatment?
Post-treatment, it violently exploded to 180 cells per square micron, an 18-fold influx of fresh recruits flooding the battlefield.
It's incredible. But the predictive power here isn't what happened after treatment. The crucial clinical finding is what that staging area looked like before the first dose was ever given.
Oh, wait, really? Baseline matters that much?
Immensely. Patients who had a high proportion of GZMK-positive cells physically waiting in the stroma in the parking lot before treatment had a significantly higher pathologic response. Their tumors essentially melted away from the immunotherapy.
Wow. Because their staging area was already heavily stocked with the right troops.
Exactly. The immune system had mobilized the fresh progenitors. They were just paralyzed by the tumor's chemical brakes. When the neoadjuvant therapy removed those brakes, the GZMK cells immediately crossed from the stroma into the parenchyma, differentiated, and cleared the cancer.
But if the parking lot is empty at baseline,
then removing the brakes achieves nothing. You can't unleash troops that aren't there.
That makes so much sense. Okay, recap time. Bullet 1. The stroma is the staging area, and the parenchyma is the core battlefield. Bullet 2. Treatment causes an influx of cells from the stroma to the parenchyma. Bullet 3. Having a high baseline of GZMK-positive cells in the tumor's stroma before treatment strongly predicts if the therapy will work.
Spot on again.
So thinking about that, here is a question to you. If we know these cells predict success in head and neck cancer, could this be a universal rule for all cancer?
Well, the researchers wondered the exact same thing. Does this reflect a fundamental pillar of human immunology? Can we scale this up?
And they absolutely went for it. They didn't stop at 11 head and neck cancer patients. They engineered a way to scale this up by creating a 40-gene expression signature based on these specific GZMK-positive commuter cells.
This was a brilliant move because you can't practically do single-cell sequencing on every patient globally. It's too slow and expensive. So they built a mathematical score, this gene signature, that can be applied to standard bulk RNA data.
Right, taking the smoothie of a standard biopsy and mathematically extracting the specific flavor of the GZMK pathway.
Exactly.
And the evidence levels here are just wild. They tested this against a who's who of global cancer cohorts. The TCGA cohort, the FOI cohort, the IM-Seasion trial. And it wasn't just head and neck cancer anymore.
No, they broke completely out of that limitation.
Yeah, they successfully predicted immune checkpoint blockade response in melanoma using the GID cohort, non-small cell lung cancer using the DOOM cohort, liver cancer in the SUS database, gastric cancer with the KIM data, urothelial cancer with the Moriartyson cohort.
What's fascinating here is the sheer biological conservation. Across radically different organ systems and mutation burdens, this single GZMK signature accurately predicted who would survive.
And it even beat the existing heavyweights, right?
Absolutely. They benchmarked it against the established giants, the cytotoxic signature, the interferon gamma signature, the TLS signature. This new 40-gene GZMK signature achieved an average AUC and area under the curve accuracy score of 0.79.
Okay, an AUC of 0.79 across that many independent cohorts is staggering.
It really is. In the messy world of human biology, it systematically outperformed the clinical standards. It proved that the biology of these specific T cells is a fundamental pillar of human cancer immunity.
Okay, final recap of the deep dive. Bullet 1. The researchers created a 40-gene mathematical signature to detect GZMK cells in standard biopsies. Bullet 2. They validated this across massive global cohorts for melanoma, lung, liver, gastric, and urothelial cancers. Bullet 3. The signature achieved a massive 0.79 accuracy score, proving it as a universal biomarker.
A true master key for diagnostic prediction.
So, question to you. We've gone from the microscopic diary of a single cell to a tool that could change global oncology. Are we entering an age where your immune profile is more important than the tumor's profile?
I really think we are.
So what does this all mean? We journeyed from understanding the toxic tumor swamp to tracking individual cell clones with GPS-like precision. We uncovered the secret weapon of GZMK positive commuter cells, mapped their staging grounds in the stroma, and finally validated a 40-gene signature that predicts survival universally.
And it leaves us with such an exciting, provocative thought for the future. If baseline stromal density is the absolute bottleneck, if a patient lacks these GZMK progenitor cells, how might we engineer them?
Oh, wow, like inject them.
Yeah. Could we harvest blood, engineer these commuter cells, and artificially stock the parking lot before delivering the immunotherapy? We could literally guarantee the staging ground is ready.
Engineering a non-responder into a guaranteed survivor. That is incredible. Well, thank you so much for joining us on this deep dive into the front lines of precision oncology. It's a reminder that the key to curing disease is often already written inside your own cells. Catch you on the next one.